Tissues. Everything in life is built in a hierarchical way. Biology is concerned with organisms. We are organisms, right? And even if you are interested only medically, you are interested in the health of a particular unique individual organism. Uh, and organisms are made of parts. Now we are not made of one type of molecule repeated a million times and that's it. We are not made of stone, so we can chip a little bit here and there, and what's left is still me. No. So there is a hierarchy of organization, and every level of organization looks different and is organized in a different way. So organism is made of a number of organ systems, and as we talked about it before, it's really fuzzy what the borders between those systems are because pretty much every cell in the body is part of multiple systems. Those systems are made of organs, and the organs are made of tissues. Tissues are made of cells. Cells are made of organelles, and organelles are made of molecules. Molecules are made of atoms. So tissues are somewhere in between, somewhere in the middle of the hierarchy between molecules and organisms. They are the ways that the body is organizing its cells. So you are going to, during embryonic development, a whole bunch of different cell types made, and those cell types are not going to be in random places. They're going to be organized in tissues, in groupings of cells, usually made together and sticking together. There are four different types of tissues. Those are the epithelial tissues, connective tissues, muscular tissues, and nervous tissues. Now, muscular tissues are very specialized, and we're going to talk about them in detail when we cover the muscular system. Nervous tissues are also very specialized, and we're going to talk about them when we are covering the nervous system. So let's talk just for a few minutes about the two generalist kind of types of tissues, which are epithelia and connective tissues. Epithelia are tissues in which cells cover surfaces. Connective tissues are all the others that are not covering surfaces, they're between the surfaces. So, epithelia are going to have cells that are densely glued to each other to produce sheets, and those sheets are then covering the surfaces. The surface can be on the outside, the skin, right? That's the surface, obvious surface. But there are other surfaces on the inside of the body. There's the surface of my nasal cavity, and surface of the inside of my mouth, and the surface of the inside of my heart and my arteries and veins, and the surface of the inside of my intestinal system. All of those are surfaces, and all of those are covered by epithelial tissues. There should be no breaks, no holes. Those surfaces should very clearly delineate what is above them, what is under them, what is the outside world, what is the inside world. And so they're going to be densely populated, densely glued to each other. And we, as humans, we like to classify stuff, so epithelia can be classified. They can be classified according to a couple of different criteria. We are going to go into great detail of a bunch of them, mainly in the lab, a little bit in the lecture. This is mainly coming straight out of the textbook as far as your studying goes, but let me just go over just the basic criteria for classifying epithelia. One is, are those cells organized in a single layer or in multiple layers? So is it going to be a simple epithelium, or is it going to be a stratified epithelium with multiple layers? Uh, Another way to classify is by the shape of cells. So cells that are very, very flat, they are squamous. So there will be a squamous epithelium. If the cells are about cuboidal, which means the shape of a cube, which means the height and the width are about the same, there's a cuboidal tissues. And then if the cells are much taller than they're wide, so they're really, really kind of spindly tall things, those are called columnar tissues because they're making a column. And so you combine those two types of classifications, and you're going to get things like simple squamous, simple cuboidal, simple stratified, and you're going to have columnar. You're going to have stratified squamous, stratified columnar, stratified cuboidal. And we are going to, especially in the lab and looking at the slides, we are going to identify all of those tissues and where they belong. Where do you find different types of tissues? Which ones are very commonly found in our bodies? Which one are rare in our bodies? So, for instance, simple squamous epithelium is the epithelium that covers the insides of our blood vessels, like capillaries. So, very thin cells, just one layer of cells. So, it's relatively easy for various small molecules and gases to pass through. The alveoli in our lungs are made of simple squamous epithelium. Again, a very flat, very thin layer, a single layer of cells, so gases can pass easily across the membrane. All of these cells are always attached to a basement membrane, which is a very, very thin membrane made of some proteins and carbohydrates, an interesting mix. Underneath it, it's usually some kind of a connective tissue. But epithelial tissues are going to be on top of the basement membrane. Uh, the simple cuboidal, found, for instance, in the nephron in the kidney. Uh, uh, the simple cuboidal is relatively rare in the human, uh, but we can find it, for instance, in the intestine, lining the intestine. Then stratified squamous. We can find it in esophagus. If it's many, many layers and top layers are dead and kind of feel, you know, kind of sloughing off, that is the example of the skin. That is a keratinized epithelium. Then stratified cuboidal, I don't think can be found in the human, and the stratified columnar is very rarely found in the human. We're going to look in your lab manual for maybe some exceptions. There are two epithelia that don't belong to any of this. They are unclassifiable. We don't like when we can't classify everything perfectly. One of them is ciliated pseudostratified columnar epithelium. If we dissect this name, which contains four words, we can kind of figure out what it is and how it looks like. First, well, it's epithelium, so it's covering a surface. So those cells are bound to a basement membrane. Um, it is columnar, which means that the cells are tall and narrow. It is pseudostratified. What does that mean? What it means is that when you first look under the microscope really quickly, it looks like it's many layers. It looks like it's stratified because the nuclei are at very different levels. So it seems like there's multiple levels of cells. But then you look a little bit more closely, you put it in a higher magnification and look really close, and you notice that every one of those cells is bound to the basement membrane. Every one of them goes all the way to the bottom, which means there's only one layer. They just give an optical illusion that they're in multiple layers, but there's only one layer. Ciliated means that they have cilia at the apical surface. What is apical surface? That's the surface that's the opposite of the basement membrane. So the tip of the cell is the apical surface. The apical surface has cilia. If you look at ciliated pseudostratified columnar epithelium, you're going to notice that every now and then you're going to find something, a cell that looks a little bit different. That's a goblet cell. It's a secretory cell that secretes mucus. and then mucus gets secreted onto the surface, that's apical surface of all these cells where all those cilia are. And so all those cilia are moving, they're beating, and they're moving the mucus. So where do you think you would find something like this? Because form follows function, there is a particular place in the body where you really, really need this kind of epithelium. And this is in the respiratory tract. So nasal cavity and the throat and the trachea and the bronchi, they are covered on the inside. Those surfaces are covered on the inside by the ciliated pseudostratified columnar epithelium. And what do? They are going to trap all sorts of things that are coming in with the air. We are not breathing perfectly sterile air. Air that we breathe in is going to contain dust, smog, smoke, pollen, bacteria and viruses, all sorts of stuff. Gunk and goo is going to come in with the air, and the air is going to come into our nostrils in a laminar flow. Laminar flow means that all the elements, all the molecules, all the dust particles, everything is flowing in the same direction at the same speed. And then they come into our nasal cavities and they're ridges. And we're going to talk about those ridges when we talk about bones. And then in the next semester, when we go to the respiratory system, those ridges are called nasal conchae. And in between them, are kind of ditches, which are called nasal meatuses. But there's bony structure underneath those. Those are the turbinate processes of the ethmoid bone, and these processes are going to make it very convoluted inside of the nasal cavity, and suddenly that laminar flow of air is going to be broken, and you're going to have turbulent flow of air. So all these particles, gas molecules, everything is going to start going every which way, and is going to get stuck in the mucus. And you're breathing in, and it goes all the way through the trachea and the bronchi, and it continues being turbulent and continues getting stuck in the mucus. And that mucus is embedded right there where the cilia are on the apical surfaces of those cells. And those cilia are slowly moving. They're beating, but they're beating in a particular direction. They are beating up towards the throat, towards the larynx, if they are coming from the bronchi and trachea, or they're beating downwards from the nasal cavity and the larynx. So you are going to end up with all of that gunk and goo stuck in the mucus. It's all going to end up in your throat. And once it's in your throat, this is the part where we have skeletal instead of smooth muscle. This is the part of the respiratory system that we have conscious control over, which means that we can decide what to do with all of that gunk and goo in all that mucus and all the dirt that came in with the air and then got stuck there. We can spit it out, we can cough it out, we can swallow it and throw it into the hydrochloric acid of the stomach for it to be dissolved. Uh, and this prevents the air from bringing all that gunk and goo, all the dirt and pollen and viruses and bacteria all the way into the alveoli. Because if it gets to the alveoli, there's very little that can do. There's so-called dust cells or macrophages there that are scanning the inside of the alveoli, trying to catch if there anything went through. But if not, you're going to get a bronchitis or pneumonia. You can get killed by toxic gases. The easiest way to kill is through gases, through the toxins that are gases. That's the easiest way to kill an organism, kill a mammal or a bird. Especially a bird, because they have efficient lungs, which is why we put canaries into coal mines, because they're going to die before we do, because their breathing is more efficient than ours is. There's one more spot where you find a ciliated pseudostratified columnar epithelium, and that's about maybe a half an inch to an inch long part of the urethra as it passes through the prostate in males only. This is a place where you have two vas deferences fusing, the sperm and semen are coming in right there. And so all of that kind of gunked up semen needs a little push, needs a little help to go through to the rest of the urethra. So we have cilia there as a part of ciliated pseudostratified columnar epithelium. There's another epithelium that is not classifiable, and that is transitional epithelium. Transitional epithelium is found in the ureters and the urinary bladder. So let's say you just went to the bathroom and your urinary bladder is empty. Your wall of the urinary bladder is going to be pretty thick. It's going to have 8, 9, 10, 11, 12 layers of cells. If you look under the microscope, they're kind of circular, more than cuboidal, and they're all over the place. And you can kind of figure out, well, there's, let's say, 9 layers of those cells in the wall. And then, the urine is coming from the kidneys, dripping little by little through the ureters into the urinary bladder, drip, drip, drip. And as it's dripping, it's starting to fill the urinary bladder. And that liquid, as there's more and more of it, is going to exert pressure onto the wall of the urinary bladder and is going to start expanding it. So the way the wall expands, it stretches. What does it mean to stretch? Those cells start moving on top of each other, and instead of 9 layers, now I have 8, and then 7, and then 6, and 5. Now you're starting to feel it. 4, you definitely need to pee. 3, you're running to the bathroom. 2, 1. 1 is possible to be done in a lab. You can actually take the urinary bladder and bring it all the way down to a single layer of cells. Uh, and then there are all these muscles. In next semester, you're going to learn how it all works, how the urination works. There are all these muscles they're going to squeeze. And what is going to happen is that the cells of the epithelium are going to start moving back on top of each other, and they're reconstituting the 9 original layers. This process is a positive feedback loop. It cannot be reversed and stopped. Once you start peeing, you can stop for a second, but you have to finish. You cannot just like, oh well, I changed my mind. I peed a half, and then I'm going to go with my life, and maybe I'll pee the other half later. No, once you start peeing, you have to finish because it's a positive feedback loop. It cannot go backwards once it started until it reaches its goal, which means that transitional epithelium has its own positive feedback loop. So we're constituting the maximum number of layers out of the situation where it has minimum number of layers. So that's an unusual type of epithelium. Now let's go to the connective tissues. Connective tissues. Again, we're going to talk about a lot of them uh, in context of different organ systems. We're going to talk about blood and bone and teeth and all those other tissues. But in general terms, the connective tissues are found underneath the basement membranes of various epithelia. They're filling up all of that space. And they are going to have 3 elements. They're going to have cells, they're going to have fibers, and they're going to have matrix, an organic matrix, which is just a bunch of molecules. And they range from completely liquid through kind of soft to harder, harder, harder to very hard, very solid connective tissues. So, what would be the liquid connective tissues, the connective tissues with the liquid matrix? Those are blood and lymph. If you are talking about, let's say, something that's a little bit more solid but still soft, let's say adipose tissue, reticular tissue, that's kind of in the middle. Then you get harder and harder, and you're going to get to things like tendon and ligament, which is going to have much less and much harder matrix and much more, for instance, fibers. And then at the other end of the spectrum are the hardest possible and most solid connective tissues. Those are teeth, dentin, bone, and a little bit softer than bone is cartilage. And we're going to talk about these when we talk about the skeletal system and whatnot. So uh, just remember that there's this whole spectrum. So, how many cells, what kinds of cells is going to be different? How many fibers, what kinds of fibers are they going to be? Elastin, are they going to be collagen? It's going to be different. For instance, blood and lymph, they don't normally have fibers, but they have potential. Those cells have a potential of making fibers if needed. For instance, when you need to make a blood clot. Um, so fibers are possible, but not always present. And then you have matrix that can be made of all sorts of different kinds of molecules, and it can be ranging anywhere from completely liquid to completely solid, and anything in between. So we're going to look at a variety of those tissues in the lab once we get to the tissue lab second week or so. And we're going to look at different types of connective tissues, and we are going to talk about each one of them, where it is found, what is its function, how does the organization and orientation of cells and matrix and fibers give it the function that it needs. So I'm going to stop here, and we are going to talk more about that in the lecture and in the lab.